Reproducibility. Is there a crisis? Irreproducibility is when there is a failure to repeat an experiment and/or confirm the original data. This is a growing concern. One of the first studies addressing the crisis by John Ioannidis claimed that most research findings are false. The next prominent study in 2008 looked at treatment descriptions in 80 papers. 51% did not have sufficient detail to be replicated. Then, in 2011, Prinz et al. Repeated 67 of their own studies. They found that only 25% could be reproduced sufficiently. In 2012, Begley and Ellis looked at 53 landmark studies. They found that only 6 could be reproduced. Later that year, in a separate study, it was found that out of 258 publications, only 127 results could be replicated. In 2013, Valinsky et al. Looked at 238 published studies to determine if they could identify the exact materials used. For example, cell lines, antibodies, knockdown reagents, constructs, and model organisms. 54% of resources could not be uniquely identified. As we can see, issues in reproducibility is not a new thing. The studies based on the sufficiency of method and resource reporting found, on average, lower irreproducibility estimates, whereas studies based on the exact replication of the original findings showed averaged higher estimates. This gives a range from 50 to 90%. These papers and others were collated by Friedman et al. In 2015. Using a 50% irreproducibility level, they estimated a cost of $28 billion per year, wasting time, resources, and slowing progress of research. Issues in reproducibility affect many other areas of science. Four main causes have been identified. The most common is biological reagents and reference materials at 36%, followed by study design, data analysis and reporting, and lab protocols. Examples of reagents and materials include cell lines, animal models, and antibodies. Starting with cell lines. Accurate documentation of species, sex, and tissue origin is essential for the reproducibility of results. Misidentification of cell lines was first reported in 1968. The authors estimated that 20% of cell lines are misidentified. This equates to over $700 million in research that could be at risk. An example of misidentification was the incorrect characterization of a breast cancer cell line, which was used in over 1,000 publications. These were invalidated when it was later found to have an identical profile to a melanoma cell line, decreasing the validity of results found using either cell line. Results from a recent survey looking into the main causes found that 30% did not receive authentication training. 33% were found to share cell lines between labs. 67% did not verify their cell lines. Now looking at animal models. These are often argued to be poor predictors of human disease and their therapeutics. However, often the design and choice of animal model lacks scientific rigour and robustness, which contributes to the high failure rate. In a 2009 study, a mouse model was reported to display the key motor symptoms of ALS. The model was used in preclinical drug development studies which progressed to advanced stages of clinical trials. These ultimately failed, resulting in loss of hundreds of millions of dollars. It was later discovered that this was a poor genetic and phenotypic model of ALS, thus demonstrating the need to fully validate animal models. Moving on to antibodies. Glenn Begley said, "Poorly characterized antibodies probably contribute more to the problem than any other lab tool. The key issues involving antibodies are batch variation, cross-reactivity, and undefined dynamic range." An example of hindering research is by Grimsey et al., who tested CB1 receptor antibodies from various commercial sources in HEK cells. used Western blot and immunofluorescence and found that only 2 out of 6 antibodies showed specific staining. Efficient and reliable research requires sensitive, specific, and well-validated antibodies for the protein of interest. The antibody market is worth $1.6 billion. More than 300 companies sell over 200 million antibodies for research. This has been estimated to represent 250,000 to 500,000 unique core antibodies. In 2011, Eaglehoffer looked at 246 antibodies and found that a quarter failed specificity tests, and 4 were perfectly specific to the wrong antigen. This clearly highlights the big issue in the antibody industry. Data bias is another big cause of the crisis. Publishing fake results is relatively uncommon. However, unconscious bias during analysis and reporting of data can lead to false positive results, where the null hypothesis is incorrectly rejected. P-hacking is the analysis of data often without a predetermined hypothesis until non-significant results become significant. Although unintentional, there are many ways this can occur. For instance, gathering a wide range of data in the hopes of finding a significant result without a clearly defined hypothesis. Ending data collection once a significant result has been found, rather than continuing the investigation to a predetermined endpoint. Disregarding data that opposes the desired conclusions. When publishing data, only showing part of the dataset can misrepresent the findings of the study. Selecting a single result that best represents the desired conclusions further contributes to the reproducibility issues. Hence, although the data published is true, its analysis and presentation can be misleading. Underlying these causes are constraints on time and funding, a pressure to publish and be highly cited, pressure to publish groundbreaking research to positively impact industry and health. There have been many solutions suggested to increase reproducibility. There should be a movement towards increasing transparency within scientific research. Many major journals are now implementing prerequisites of studies prior to publishing. Another solution involves proper validation of materials used in research, along with registering and sufficient documentation of their sources. Education in rigorous study design, appropriate statistical analysis, and awareness of the need for validation should be an integral part of research training. Implementing these changes will have a high upfront cost in terms of both money and time, but should be outweighed by the long-term benefit. To summarise, in research, efficiency of 100% and waste of 0 is neither achievable nor desirable. However, there is room for improvement to increase reproducibility to an acceptable level.